Okay, well, hello again, everyone, and welcome. Today we have Mrs. Lorenza Allen, Technical Marketing Specialist for Enartis, who is going to present over tannins and winemaking. So without further ado, Lorenza, the floor is yours.
Thank you, Aaron. I'm going to share my screen. Can everybody see the presentation?
We can see.
Perfect. Okay, so talking about tannins in winemaking today. This is, it's not going. Okay, there we go. This is some of the points that I would like to touch today. I will give a quick introduction about tannins, in which forms they're found in nature, how the production process for enological tannins works, and the different types and why it's important to know the differences. And then we'll get to the juicy part and how to use tannins in winemaking and the different applications. So what are tannins? Tannins are found in nature, and they're like antibodies for plants. They have the function of protecting plants from external aggressions. They are known for their bitter and astringent taste that repels animals from feeding on the plants or the fruits prematurely. They're also very important for, in case of viral, bacterial, fungal infections. They can slow down the growth of pathogens and limit the oxidation, so the deterioration of the tissues of the plants. In nature, they're found in trees, in shrubs, and they are structured from the leaves, the fruit, the seeds, the skins, and the wood. In winemaking, the plants that we use mostly for the production of tannins are oak, tara, gall, chestnut, mimosa, quebracho, and obviously grapes. Tannins in nature, they differ in composition and chemical structure, and we'll have a quick look at why it's important to know. So how are tannins made for enological use? The process is relatively easy. We start from the raw material, so the matrix, it can be a type of wood or a fruit or so on, a solvent, most often water. And then these two go through an extraction process. So the aim is to separate the solid from the liquid. And there are three factors that affect this extraction process: the time, the temperature, and the technique, which can be maceration, percolation, cavitation. At the end of this process, we end up with two products. One, the refined exhausted product that will be discarded. And the other one is the tannin solution, which is the one that we're going to use. The tannin solution, then basically imagine it as if it was a tea. We basically made a tea of tannins. And then we have to concentrate this tea. So we're making a concentrated tea so that there are more tannins for a quantity of liquid. After that, it goes through atomization. So we extract the liquid so that we only have the solid of the powder by evaporation. In this process, temperature is critical and it's very important. We try to keep the temperature as low as possible, but still having water evaporate. And we know that quite intuitive that the higher the temperature during this process, the more likely or potentially is the lowering of the quality of tannins. So when we have a high quality tannins, or also the most expensive ones usually, those are extracted at very low temperature, around, let's say, 90 Fahrenheit degrees. After that, we are left with the solid part, a powder, and we go through the granulation process. This is a process that is not extremely important for tannins as it can be for other products, but it's still important to optimize the ratio between the powder, so the solid part, and the humidity. This makes it so that it's easier for the tannin to dissolve in water. So when we add our tannin to our solution, water or wine, it does dissolve. That's why it's important. And then we have different types of tannins. We have many different types of tannins, but we divide them in two main categories: hydrolyzable and condensed. And then from this we have more. They are distinguished because the condensed ones are oligomers or polymers of flavan-3 oils. They're also called proanthocyanidins, and they release anthocyanins by hot acid hydrolysis. The hydrolyzable ones are gallic and ellagic tannins, and they consist of a sugar, usually glucose, that is esterified with either gallic acid or ellagic acid. They come from different raw materials, and sometimes, because some of them, with my pointer out, where is it? Here. Okay. Some of them, like these ones, they're very hard to pronounce and no one wants to remember the names. We just refer to them as the material, the tree where they come from, like Bracho and Mimosa. I wanted to show you a little bit of the chemical composition. I will not go into extreme details of the chemistry of tannins. It's not the aim of this presentation, but I think it's important to know how they look like, because their size and dimension really influence the role that they have in winemaking. So in this case, when we talk about condensed tannins, it's a repetition of the same molecule over and over, very well organized. And then for the hydrolyzable tannins, so gallic and ellagic tannins, they have, in this case, ellagic acid and the gallic acid attached to a molecule of sugar. So one thing to know about tannins is that they are all phenols. This is important because of the aromatic ring and the hydroxyl group, so the OH group. So let's start with the aromatic ring. They have charges that can move around the ring, so they can move in both directions, and that influences their ability to react with other charges from other molecules. And then the OH group influences their ability to, for example, condense and be a nucleophile. And this has effect on how antioxidant or anti-radical the tannin is at the end. So these properties, the shape and the size, really are important to understand how to use a tannin. This is just to give you a quick look of how the molecules look. So we have seen that this more organized type of tannins, the proanthocyanidin, and then we have the gallic and the ellagic. So the gallic, they have the sugar in the middle, usually is glucose, and they're bounded. So the aromatic ring, the gallic acids, they're all connected individually to the sugar. And then the ellagic tannins, they're all connected to each other, so there are more bonds between the rings, and this makes it so they are more anti-radical compared to the gallic tannin. Now to the more interesting part, how to use tannins in winemaking. So when we talk about tannins in general, we know that they are plant extract, that they are famous to be astringent in taste or bitter, and that they precipitate proteins. But there are a variety of other things that the tannins can do, and the way, their composition and also the way they are extracted significantly influence their effect in wines. All tannins have all of these effects, but some have more than others, and that's why we select them for their main purpose. Starting with proteins interactions. So this is one of the reasons why tannins have been used in winemaking since the beginning. Their ability to interact with nature, with proteins in nature, makes it so that they have an antimicrobial action towards pathogens in the plant. In winemaking, they interact with proteins, and they have two main goals. One is to protect color, The other one is to remove unstable proteins. So this is very important, especially at the beginning of the vinification in must, when we can remove some of the unstable proteins and avoid a haze later on. And we can also remove some of the proteins that will interact with color and therefore save some of the color. So the tannin will precipitate with the protein instead of having the color precipitating with the protein. And the reactivity here really depends on the type of tannin. So in nature, we have almost an infinity of different tannins because of the huge variety of proteins that are found in nature. So the way a tannins interact with the protein really depends on the protein. For example, we know that astringency is one of the things that we know about tannins is because of the interaction that the tannins have with the proteins in our saliva that gives us the astringent taste. But that doesn't mean that a wine that is particularly astringent will result in being more reactive with unstable proteins. So for example, thaumatin, which is a very unstable protein found in wine, reacts very well with chestnut, but chestnut doesn't give us an astringent sensation when we drink a solution. Then tannins also have antioxidant, antioxidazic, and anti-radical properties. These are very interesting and slightly different from each other. So when we talk about antioxidant, is the ability to block an oxidation reaction, which involves oxygen, but oxygen doesn't really oxidize the wine unless there are metals involved, especially iron and copper. When we talk about antioxidazic reaction, is the fact that tannins have to stop the negative effect of oxidative enzymes, so an oxidation that will happen because of enzymes in the solution. Some of these enzymes are, the most famous are lactase and tyrosinase. Lactase is the ones that comes with botrytis, so they're both oxidative enzymes. And what the tannins, when we add tannins, what happens is that especially hydrolyzable tannins can limit the action of this enzyme. So in this case, when we talk about oxidase, if oxidase looked like Pac-Man and the mouth was the active site, tannins will basically block the active site so that the lactase will not be able to react with other molecules in wine. Same for when we talk about anti-radical. Tannins can, and we will see this in a few slides, they have the ability to chelate metals. So they remove or they bind with metals, blocking the action of aggressive molecules, so the free radicals. This is a comparison between the antioxidazic effect of sulfur and tannins when we talk about, again, lactase and tyrosinase. So the red line is lactase, the blue line is a tyrosinase, and we can see that the activity by percentage slows down or is reduced as we increase the amount of sulfur. And basically to reduce a lactase by, let's say, around 60%, we have to use around 70 ppm of sulfur. When we do the same with tannins, let's say gallic plus ellagic tannin, we can have the same result by using 20 grams per hectoliter of tannins. This makes it so that we can manage our sulfur protocol differently and either add less sulfur or know that the sulfur that we use does not get all used up with this type of activities. Similarly here, we're talking more about the antioxidant effect. So this was an experiment done by the University of Asti in Italy. And it's a wine, the same wine bottle with the same amount of sulfur, and then the amount of free sulfur found in the bottled wine after a couple of months. The blue line is the wine as it is, only with sulfur without tannins. And then in the others, we had an addition of Tara tannin and 2 different oak tannins. And we see that at the end, there is a decrease, significant decrease in the control, significant decrease of free sulfur, while the tannins, the samples that were treated with the tannins were we see a slight decrease, but much less significant than the one without tannins. This is another similar effect. This is an experiment or a trial that we have done internally using two different tannins, Hideki and 10-SLI. They are both blends, and we call them technical tannins. And we had a bottled wine with a 0 free SO2, and then we measured the optical density increase in the control. There was the one without tannins and the other two, and we see that if we add tannins, the percentage is very limited, so there is less antioxidant effect and more protection from the tannins. Talking about the anti-radical effect of tannins, this is a trial that was done by Vivas in France. They test the protection of wine from oxygen. So this test is done by adding hydrogen peroxide in the presence of tannins, and then we see how much of this hydrogen peroxide is left at the end. So if a lot or all of the hydrogen peroxide is still present at the end of the test, it means that the tannin that was in the solution does not have an anti-radical activity. So here we see in gray the control that didn't have any tannins, and at the end of the test is still 100% of the residual activity. And we see that if we add tannins, this activity is much lower. So especially the gallic and the ellagic tannins, they are the one that shows the best anti-radical activity, or they are most effective against radicals. Again, we mentioned before metal chelation. This is an important property of tannins, and it works in two ways. One, against the oxidation, and also the other one against the microorganisms. So as mentioned, copper and iron are both involved in the oxidation process, and they are also responsible for the formation of free radicals. And then the other one is metals like, again, copper and iron, but also magnesium, zinc, and so on. They are cofactors for the function of many enzymes that promote the growth of microorganisms. So, but while if we use tannins, we eliminate the presence of these metals, and therefore we stop the oxidation process of the growth of microorganisms. So here we have an example, and the most reactive, or the one that can chelate, in this case copper the best, are the gallic and the ellagic tannin. Tannins also have an effect on the antimicrobial effect. This is something that has been known since the beginning of the 1900s, when we started using tannins in winemaking. In fact, we can find old enology textbooks that talk about tannins as antiseptic, and they can be, they are recommended for the treatment of unhealthy grapes during harvest in order to stop or control the development of unwanted microorganism or bacteria. In this case, this is an experiment that we have done with Hideki, again, one of our technical tannins, and the control of acidic bacteria. So we have a control with no tannins, and then the control plus 5 grams per hectoliter of tannin. And we see that after around 3 months, the control sees a spike in acetic acid. So most likely there is an acetic bacterial growth in the wine, while the sample treated with Hideki basically has the same amount of acetic acid over time, meaning that the tannin is able to control the growth of acetic bacteria. Similarly, with tannin, we can also control or manage the growth of malolactic bacteria, so the malolactic fermentation. This is a trial that we have done using the synergy between this technical tannin, Hideki, and step micro M, which is activated chitosan. Activated chitosan is a product that removes unwanted microorganism. So in this red wine, pH 3.6, low sulfur, we treated first the wine with step micro M to remove the microorganisms. And then we added 5 grams per hectoliter of Hideki, and we had a control without tannin addition. And we can see that after, again, 2 or 3 months, there is a spike in lactic acid in the control, meaning that most likely the wine is going through or has gone through malolactic fermentation, while the wine treated with Hideki stayed at the same level, basically. So there was no growth of the bacteria. When we talk about malolactic fermentation, always important to remember that the factors that are also influencing the onset of the fermentation are pH, free sulfur, which is also influenced by the pH, temperature, and alcohol. So when we think about Hideki, we can think, or technical tannins that has these properties, we can think of them as an additional factor. So there needs to be a synergy of all of these factors to make sure that the malolactic bacteria don't take over. For all of the reasons that we just mentioned, tannins can be used to decrease the sulfur use, or at least manage the sulfur protocol, because they can be used, as we've seen, to limit the oxidations, but also because tannins can react instead of sulfur. So, for example, if they can react with hydrogen peroxide, and they can regenerate ketones. So if we have tannins in the solution, they will basically save sulfur, and at the end you will have a higher amount of free sulfur than you would otherwise. Below is a chart that we've done in bottled wine with the same technical tannin. Testimonies in Italian means control is the blue line, and then we have 2 other samples, one with 1 gram, the other one with 3 grams of technical tannins, which are pretty low dosages. And we've seen that after 4 months after bottling, the content of free SO2 is higher in the samples that have been treated with the tannin instead of the control. It still decreases, but we're having a higher free sulfur at the end, which is significant. And again, the dose of tannins here is pretty low. Moreover, very important, color stabilization. Tannins are one of the main factors in color stability in red wine. And this depends on different things. On one hand, the ability of tannins to consume oxygens, which prevents the oxidation of the tannins, but also their ability to combine directly with anthocyanins and to create pigments. So, for example, when we talk about anthocyanins protection, as we mentioned at the beginning, tannins can remove proteins. They can bind and precipitate the proteins that will otherwise bind and precipitate the color. So we have a higher color. And they also decrease the polyphenol oxidase. So we have less color oxidation. This is all very important, especially in must. And then we have other reactions like condensation and copigmentation. When we talk about copigmentation, this is also very important in young red wines. And it's the formation of complexes between anthocyanins and cofactors like flavonols in general. And so the tannins here, through the copigmentation, they protect the pigments from the oxidation and limit the color loss. And then when we talk about condensation, this is when we form, or pigments are formed via a direct bonds between anthocyanins and tannins in oxidative condition. When we talk about acetaldehyde bridge, this is the case. And this is something that happens in wines that are destined for longer aging. So wines that will be consumed when they're not very young. And this can be done also with microoxygenation. When we talk about microoxygenation, this is the reaction that happens. And having a tannin here really helps this reaction and the condensation, so the formation of these pigments. In the graph below, here it shows the color intensity of wine. So the sum of the 420, 520, and 620. So the hues of yellow, red, and blue colors in wine. These are 2 samples treated with a mix of tannins. I think it was a pretty low dose, like 1 grams per hectoliter of tannin each. And the control. So we see that even though it might be not a whole lot, you think, from less than 4 until more than 5, it's still a significant difference for the amount of color present in wine at the end of the day. And then mercaptan bindings. So mercaptans are the sulfurous compounds that are formed during alcoholic fermentation from hydrogen sulfides. And, you know, they have those smells of like cabbage, onion, rotten eggs when it's really bad. What tannins do, they bind with mercaptans and they remove these unwanted flavors, and we have a wine that is at the end more fruity. What happens with tannins is safer and better treatment than what we will have with copper, because with copper we will not have the aroma and the bouquet that we have with tannins at the end. And the way that tannins work, we have 2 different ways. One is a direct condensation reaction. So the tannins and the mercaptans react together, and the tannins remove the mercaptans. And on the other hand, we have an oxidation or a coupled oxidation. So we have the formation of hydroperoxide and the formation of free radicals that will destroy the mercaptans. So in any case, when we have this sort of off flavors, doing trials with some of the tannins that have these abilities can give great results before we use copper. And then last but not least, the sensory improvement. So we have seen that we can use tannins basically every time during our winemaking process, from as soon as the press until the very end. And I will also say that a lot of the tannins that we talked about, or the characteristics that we talked about, are for tannins that have technical purpose, but we also, or there are also the tannins that are purely used for sensory improvement. So we know that in general tannins are bitter and astringent, but we have other sensations or other attributes that tannins can contribute to wine. They can increase the structure of the wine, make the wine fresher, more elegant, more round, and also contribute aromas like oakiness, as if the wine aged in barrel, and vanilla. They can enhance the red fruits, and mask herbaceous notes. This is just some example of some of the tannins that we have in our portfolio and how they can contribute to the wine, and also how much their impact is. Some can be more impactful than others. Again, here I wanted just to show how these are all examples of different oak tannins and how they can impact the wines differently. Some of them can have more impact on the structure and the complexity, while others can impact more flavors like vanilla or caramel and soften the wine in general. When we talk about the sensory improvement of tannins, so the gustatory effect that tannins can have, it very much depends on the type of tannins, obviously, and also the dosage. So when we want to play with tannins for sensory improvement, it's always recommended that we carry out the benchtop trials before final addition. And then finally, these are just it's just a recap with some of the effects that we talked about, and our suggestions with Enartis. So we have a lot of what we call technical tannins, so tannins that don't have a sensory impact or have a very slight sensory impact, but can help with oxidation, removal of off flavors, color stability, and then there are also a lot of options for sensory improvement. And that's it from my side.